Hello, and welcome to Lab 10, Primary Production and Marine Pollution. Primary production is the biological conversion of inorganic molecules into organic molecules, such as the simple carbohydrate glucose. Organisms that perform this work are called autotrophs, meaning self-feeding. These organisms are also known as primary producers and produce their own food. In the world's oceans, phytoplankton are the most abundant primary producers. Phytoplankton is a collection of organisms such as diatoms, coccolithophyrs, green algae, and cyanobacteria. Phytoplankton are single-celled microscopic marine algae that live in the ocean's photic zone, which we will discuss later. Phytoplankton production is affected by three factors. One, sunlight, which varies with latitude, time, and water clarity. Two, nutrients, such as the availability of phosphates and nitrates. And three, the rate of predation by consumers such as zooplankton and other heterotrophs. Again, phytoplankton are autotrophs, or primary producers, and they produce their energy through the process of photosynthesis. Photosynthesis is the process of using solar energy to convert carbon dioxide and water into carbohydrates and oxygen. Photosynthesis. The following is a simplified chemical formula for photosynthesis. In photosynthesis, sunlight is required to sustain the conversion of reactants, carbon dioxide and water, into products, glucose and oxygen. This chemical reaction forms a balanced equation where the number of elements within the reactants and products will always balance and to be equal. Secondary productivity. Secondary productivity is the transfer of energy from autotrophs to heterotrophs through feeding. Secondary productivity occurs at the level of heterotroph. Heterotrophs, meaning other feeding, are organisms which feed on plant and animal material and are also known as secondary consumers. Secondary producers. Secondary producers are also known as consumers. They are up the food chain from the phytoplankton, where dinoflagellates, or primary consumers, or herbivores, feed on phytoplankton. And zooplankton, or secondary consumers, are carnivores, which feed on the dinoflagellates. Secondary consumers are respirators. Respiration is the process of converting oxygen and glucose into carbon dioxide, water, and chemical energy. Respiration. The photosynthesis reaction discussed earlier can also proceed in the reverse direction. This is known as respiration. During respiration, the reactants, oxygen and carbohydrates, react and release energy while generating the products, carbon dioxide and water. The chemical energy released in respiration is the fundamental energy source for heterotrophs. The photic zone. The photic zone is the uppermost region of the ocean where primary production occurs through the photosynthetic activities of autotrophs. It is measured from the ocean surface to the water depth, at which light intensity is approximately 1% of that on the surface. This depth is the base of the photic zone. Below this depth, insufficient sunlight exists for photosynthesis. Thus, below the base of the photic zone, autotrophs fail to thrive. Sometimes the base of the photic zone can become very shallow. This occurs in high latitudes, where there are shorter winters coupled with lower angles of sunlight that can result in a shallower photic zone, and other factors that include the time of day, time of year, and water clarity. A shallower photic zone can significantly decrease the primary production of autotrophs. Primary production as it relates to photosynthesis and respiration. Marine organisms are either photosynthesizers or respirators. The following graph shows the relationship between sunlight, photosynthesis, and reproduction. Phytoplankton, again, are photosynthesizers. They depend on sunlight for energy and thrive in the photic zone. Within the photic zone, primary production exceeds respiration. However, heterotrophs are respirators, and the rates of respiration are not light dependent. Thus, respiration occurs at all depths and all light levels. As depth increases, sunlight decreases as well as primary production. The depth where the rate of primary production equals the rate of respiration is called the compensation depth. Below this depth, respiration exceeds primary production. The light-dark bottle method. In your lab, you will graph and interpret data from the light-dark bottle method. The light-dark bottle method measures the quantity of dissolved oxygen, or O2, within a series of light and dark sample bottles collected at various depths. This method will demonstrate how the rates of photosynthesis and respiration vary with depth over time. The light-dark bottle method. To begin, a seawater sample is collected at depth containing natural concentrations of phytoplankton, or photosynthesizers, zooplankton and other heterotrophs, or respirators, and levels of dissolved oxygen. In this initial bottle, the amount of dissolved oxygen is measured and recorded. The initial sample bottle is then split between a light bottle, which is transparent to light, allowing photosynthesis, and a dark bottle, which is opaque to light, preventing photosynthesis. However, respiration occurs in both bottles. The two bottles are quickly returned to the initial collection depth for 2 hours. The bottles are then retrieved and the dissolved oxygen concentrations are remeasured. In each bottle, changes in the quantity of dissolved oxygen were the result of the following factors. In the light bottle, because it was transparent to light, dissolved oxygen was simultaneously produced by photosynthesis from phytoplankton and used by respiration from heterotrophs. In the dark bottle, dissolved oxygen was used by respiration from heterotrophs, while photosynthesis was inhibited because the bottle was opaque to light. The net productivity, or the production of dissolved oxygen, is the net change in dissolved oxygen in each bottle over the 2-hour interval, and it is calculated by subtracting the initial bottle concentrations of dissolved oxygen from the remeasured light bottle and dark bottle concentrations. In this experiment, this process was repeated at 10-meter depth intervals, and the changes in dissolved oxygen levels are displayed in the figure on the left for each depth. Net productivity was calculated for both light and dark bottles. Gross productivity was calculated by subtracting the dark bottle dissolved oxygen levels from that of the light bottle. Gross and net productivity. In part A of your lab, the following table shows measured changes in dissolved oxygen in 10-meter increments for the light bottles, or the white boxes, which represent net productivity, and the dark bottles, represented by the dark boxes. First, you will calculate gross productivity by subtracting the dark bottles from the light bottles. You will then plot net productivity as represented by the light bottles and create a graph. Finally, you will plot gross productivity and create the graph. Next, you will determine the compensation depth. The compensation depth occurs where total photosynthesis and respiration are equal, as indicated where net productivity equals 0. Next, you will determine the base of the photic zone. The base of the photic zone is the depth where respiration exceeds photosynthesis, as indicated where gross productivity equals 0. Seasonal variations in primary productivity. The following graph is provided in your lab showing the relative changes across the seasonal cycles. Based on these data, you will examine how the different variables increase and decrease relative to one another throughout the seasons. The graph is also divided into 3 parts: light availability and temperature variations, dissolved inorganic nutrients, and the relative abundances or standing crop of 3 different types of plankton: diatoms, which are primary producers or autotrophs, dinoflagellates, which are primary consumers or heterotrophs that feed on diatoms, and zooplankton, which are secondary consumers or heterotrophs that feed on the dinoflagellates. The first graph compares levels of solar radiation to seasonal temperatures at the surface and at 70 meters. During winter and fall, temperatures are most similar and converge due to the loss of heat. During summer, temperatures are the most dissimilar and diverge due to the absorption of solar energy. It is important to note that changes in temperature depths are due to the growth and decay of the thermocline. The second graph shows the relative change in dissolved inorganic nutrient levels. During the spring, there is a noticeable decrease in dissolved inorganic nutrients. This is caused by the uptake of autotrophs, which we will discuss in the next graph. Although autotrophs or phytoplankton generate their own energy from sunlight, they also consume dissolved inorganic nutrients like phosphates and nitrates. Finally, standing crop. The third graph shows that in the spring, there is a rapid increase in diatoms. This correlates with increasing sunlight as depicted in the first graph. However, with the addition of diatoms, dissolved inorganic nutrients quickly become limited, as seen in the second graph. This causes diatom abundance to rapidly decrease. The process of population reduction due to the lack of nutrients is called nutrient limiting. Furthermore, the increase in diatoms is followed by an increase in dinoflagellates, which is followed by an increase in zooplankton. As one organism feeds on the other, reproduction increases. However, reproduction causes a lag in time behind trophic feeding responses. Temperature, biomass, and nutrients. The following figure shows the relative seasonal changes in the tropics or at low latitudes, the North Atlantic or at mid latitudes, and in the Arctic at high latitudes, as well as their cumulative temperature profiles. Each graph documents the relative seasonal changes in nutrient levels, primary productivity or biomass, and water temperatures. In your lab, you'll discuss the observed differences in seasonal changes between latitudes, specifically comparing how each location relates to temperature consistencies or differences, how temperature relates to productivity, again for each location, as well as nutrient supply. Weighting and waste. In Part C of your lab, you are required to read the Scientific American article, Weighting and Waste. From the article, Weighting and Waste, the following map and table discuss America's sullied beaches. The map correlates density populations within coastal communities with the most polluted beaches and bays. The table lists key area closures due to bacterial contamination. You will use this data to answer questions in your lab. Watersheds. A watershed is a drainage area where precipitation runoff flows into waterways. After a storm, contamination runoff frequently results in beach closures due to pollution. The article Weighting and Waste compares the differences between how runoff flows over different surfaces. For example, along the coastline, natural surfaces, such as marshes, as seen in the bottom left picture, act as a buffer or filter to waterways absorbing pollutants. However, along coastlines, many wetland areas have been developed, as seen in the bottom right picture, forming impervious surfaces, such as parking lots, roads, and sidewalks. Impervious surfaces offer no buffer or filter and allow microbial pollution to freely run off into waterways. The pie chart / bar graph diagram illustrates this relationship at 6 locations. Pollution reaching waterways increases as the percentage of impervious surfaces increases relative to natural surfaces. Thank you for attending Lab 10, Primary Production and Marine Pollution. Please address your questions to your lab TA.